Hey, it's Eric Hartman with the CRF, and I'm here on the first day of ARVO.
So my area of research is female carriers of X-linked retinal diseases, and choroideremia is one of them. And so pretty much the work that I've done has looked into the variability that we see with women who are carrying a single copy of the, let's say, CHM variant. And so we've looked at what clinical tests we can do to better understand how they're impacted. We've also looked into the more molecular mechanisms, so what happens that determines that variability at a very young age. And so we've recently received funding from the Choroideremia Research Foundation to continue that work and do more advanced analyses. And for that, we're in collaboration with Oxford University as well. So we're very excited to get that project underway.
What we've done at the Mary Tyler Moore Vision Initiative is map the barriers to progress in the disease, and one of the things that we've identified as a principal barrier, and which is common to many ophthalmologic diseases, is the lack of valid endpoints for measuring visual function. So what we've done in our first round is to design 2 clinical studies that will measure new devices that can be utilized for objective assessment of visual function and match them against the standard measures of structure of the retina in order to advance progress in therapeutics development.
And what kind of work have you been doing with CHM?
Yes, so we have been doing a lot of work using adaptive optics imaging. We want to understand how the cell structures are changing in choroideremia. We are also doing cellular level function testing called ORG in choroideremia, and also we are doing adaptive optic combined with microperimetry, so AO microperimetry, in the goal to try to understand choroideremia both in natural progression of the disease and also in choroideremia clinical trial.
What was the research that you've most recently been involved with?
So I've been doing quite a few things.
Okay.
I have been involved with the X-linked carriers through supervising Senna Gochuk, who recently finished a PhD and has also been supported by the CRF. And I've also been looking at how the visual pathway changes as a result of choroideremia and Stargardt's disease. So what happens beyond the retina further into the brain.
I'm doing genetic testing for patients with rare eye diseases, patients with inherited retinal diseases such as choroideremia, and it is my goal to apply the most novel technologies to solve genetic diagnosis in patients with choroideremia and also to solve unsolved questions in carriers of choroideremia. So I would like to use top-notch technologies, making use of long-read sequencing and multi-omics to address unmet needs in choroideremia.
Right. Choroideremia may have been the first inherited retinal disease that was first identified back in 1989 or 90 by Franz Kremer, but the technology from that point to now is even more expanding. You're saying that these new cutting-edge machines will be able to give us analysis not just in short read of the gene, but long read, right?
Yes, so we have an amazing toolkit now. We saw an evolution from short read to long read sequencing, and we also evolved from one layer of omics to multi-omics, and that is exactly what the choroideremia community would benefit from if we could introduce that and if we could address unmet needs using this fantastic toolkit of novel technologies.
I am at the booth of Conan, which is an objective field measurement tool. For those of us with choroideremia, we know how hard it is to get our peripheral vision tested. And I'm here with the inventor of this machine. Why don't you introduce yourself?
I'm Ted Mattis, originally from Canada, but now I live in Australia. And we've been working on this for 15 years. The last 6 years we've been working with Conan. And we've got it to a point now where we think it's really a viable instrument and could be a big, good utility to your community.
Yeah, I think it's, we have been searching, we all know endpoints are very important for trials. And to get something that's objective for many of us with peripheral, with choroideremia, when we're getting our periphery measured, especially with microperimetry machines, we can't see the little dots because we also have flares of little lights. So anything that can make the measurements easier, and it's a very quick procedure, isn't it? How long does it normally take?
Well, there's two classes of tests. There's kind of a high spatial resolution ones, and they take about 8 minutes, but that's to do both eyes at the same time. And then there's 3 new tests that take 90 seconds to do both eyes.
Wow.
And they don't give you quite as much spatial detail about the field, but maybe if you've got a fair amount of damage, that's what you need. And it's better off being able to do the test relatively quickly and easily. And there are no button presses, so if people are familiar with microperimetry, don't have that. Stimuli are really quite large, which is properly engineered. That means that whereas with a field test, let's say you got the 10-2 pattern, you're actually only testing about 2% of the closed field. With ours, it's closer to 90%.
Wow.
Because the stimuli are quite big, and they're easy to see, and they don't get defocused, so it's meant to be a very robust test.
So, Dr. Juliana has been a good friend of the Choroideremia Research Foundation. She sent many patients our way in order to get more information about choroideremia. And in Brazil, you were the primary doctor for the Biogen trial.
Yes,
Is that correct?
I participated in the Biogen trial, the gene therapy trial for choroideremia. We learned a lot during this trial. Unfortunately, it was not a trial that had reached the endpoint, but we learned a lot and we are ready to continue working the next trial.
Absolutely, absolutely. And your research into, you've got a presentation, is that right, tomorrow?
Yeah, I will do a presentation about a psychotherapy for people with IRD.
With inherited retinal diseases.
Exactly.
And your study looked at their mental attitudes about getting diagnosed and then also getting mobility training?
Yeah, my study is trying to be the person who has acceptance and mindfulness to deal with IRD. So I do 3 months of psychotherapy group based on acceptance and commitment therapy, 1.5 hours per session. And in these sessions, we trouble and try to do and to dialogue about the main topics of IRD in the feelings of living with IRD and the psychosocial aspects about it. So in the end, we can create coping strategies to deal with IRD and create a mobility training to these people go to rehabilitation and be more independent and have a more mobility.
And you are one of the most recent awardees of a grant through the $1 Million Bike Ride. Can you explain that?
Sure. First, let me just say thank you very much to the Choroideremia Research Foundation for supporting my team and the young investigators that are doing all this science behind our studies. I'm very grateful. So we're going to use the support from the CRF to investigate cone structure and function in patients with choroideremia, and we're developing a method to
measure cone function objectively. It's called optoretinography, and the idea is that this will inform doctors about how the cones function and track over time, and hopefully in the near future we'll be able to use that in clinical trials to show whether our new therapeutic approaches are working.
Hi, my name is Dr. Lee Jones and I work in Maria's lab. I'm a psychologist by background, and we've just started a project looking at sort of health comorbidities in choroideremia, and I'm particularly interested in hearing more about sort of the patient experience and sort of more of the lived experience dynamics of patients.
Okay, layman's term, what is a comorbidity?
Comorbidity?
Yeah.
Comorbidity is when you have other conditions alongside choroideremia.
Okay. All right.
And it's kind of trying to understand if the choroideremia gene is causing those other conditions or if they're unrelated to them.
Okay.
And as you know, we've done a lot of work looking at what we call systemic biomarkers, so changes in your blood that could point to you having other kinds of conditions, such as maybe high levels of cholesterol or a higher risk of cardiovascular disease. We found, interestingly, that choroideremia patients had high levels of serotonin, which could therefore affect your sleep. And that's something that then Lee took forward, so maybe you want to just explain what you found.
Yeah, so we've got a poster here on Thursday, but essentially we'd found that patients, so male patients and female carriers, self-reported significantly poorer sleep than sort of the general population, and so we said that could be due to the links with serotonin, but actually we found that these patients weren't any more sort of anxious or depressed than sort of the normative population. So we're saying that these sleep problems aren't necessarily linked to anxiety and depression, but actually it could be what Maria was saying there about this systemic.
So what's the nutrient that you can take? Melatonin for sleeping?
Yeah, you could do.
Okay.
I'm not going to prescribe that.
Okay, no, because I certainly wake up a number of times each night anyway. So well, that's really interesting.
But if that happens, it may be worth going to see a sleep doctor just to see what's going on.
Sure.
I mean, because it could be other factors as well, and we wouldn't want to just say that, and it isn't for everyone.
No, absolutely, yeah. Well, we greatly appreciate all the work that both of you do, and we look forward to working and pushing this research even further. So thank you.